late stage PhD student at the Centre for Youth Mental Health at the University of Melbourne. So she'll speak for, I think, 20 minutes or so, Sylvia.
Yeah.
And then following Sylvia, we'll have Sarah Manuele. So she completed her PhD earlier this year at the University of Melbourne, and she's now a postdoctoral fellow at Monash University. And I think Sarah and Sylvia, do we want to leave questions to the very end?
Yeah,
Yep.
I think
Yep.
it might
Okay.
work. Thank
Yep.
you.
So feel free to pop any questions in the chat if you think of any along the way. Otherwise, you'll be free to kind of unmute yourself and ask a question at the end. Okay, without further ado, I'll hand over to you, Sylvia.
Thank you, Sarah. And as Sarah said, I'm a PhD student at Orygen Centre for Youth Mental Health at the University of Melbourne. And today I'm very excited to share some work from my PhD project, which looks at how parent emotion socialization impact adolescent brain function. So the data from 2019 show that globally, 1 in 7 young people experience a mental health disorder, with depression and anxiety, these internalizing disorders being the most prevalent. And these numbers have escalated during the pandemic. A recent meta-analysis showed that in 2020, 1 in 4 young people experienced elevated internalizing symptoms. And when we zoom in the data in Australia and for different age groups, we can see that adolescents are being particularly vulnerable to develop these internalizing difficulties. So the question we wanted to ask is why adolescents and why specifically early adolescents? And if you are parents who have a teen at home, you probably vividly remember your adolescents, they were being on their device all the time and they get annoyed easily when you try to communicate with them. And they will probably just say, fine, whatever, when they try to initiate a connection. And if you don't have a teen, I'd like to invite you to travel back to the time when you probably just started high school. Maybe you remember you were feeling quite awkward and unsure of yourself, trying to fit in different social groups and feeling that nobody really understands you. So that kind of characterizes adolescence as a period of change. It comes with physical, social, emotional changes. And as Sarah said, it's also a time where the brain is going through substantial remodeling and development, which may help to explain some of the adolescent behaviors and adolescent experience. So I'd like to introduce you this kind of quite an influential neurodevelopmental model, which suggests that brain regions mature at a different rate during adolescence. So the limbic systems that's responsible for generating and processing emotions, they develop relatively early. That involves regions such as the amygdala, hippocampus, ventral striatum, for example. And in comparison, the prefrontal regions that's responsible for cognitive control, emotion regulation, they mature at a slower rate during adolescence. And that involves kind of the lateral and the medial part of the prefrontal cortex and also the anterior cingulate cortex. All right, so as the adolescent brain is going through all these beautiful changes, it's also very plastic and sensitive to environmental influence. And when thinking about important environmental factors, we acknowledge that during early adolescence, there is increased peer influence and there are other social environments that's important for brain development. But nevertheless, parents still plays a very influential role in shaping adolescents' understanding and regulation of their emotions. Okay. And in particular, I want to introduce the idea of parent emotion socialization. I will describe this in a little bit more detail later, but broadly, it refers to how parents role model, react to, and discuss emotions with their teens. And neuroimaging studies show that parent emotion socialization is associated with adolescent brain function during emotion regulation. However, when we look into the literature, evidence has been quite inconsistent. So I will just take two examples that look at amygdala activity, for example. So for the study on the left, we can see that unsupportive parent emotion socialization behaviors was found to be associated with increased amygdala reactivity, while the study on the right shows that unsupportive parent emotion socialization was associated with decreased amygdala reactivity. And the mixed findings have been reported in other brain regions as well. And compounding with the issues of mixed findings, existing studies have only looked at correlations. We are not sure whether there is a causal relationship, such that whether parenting has a causal influence on adolescent brain function and brain development. To test causality, it's important to implement a randomized control trial design, which will allow us to experimentally manipulate parent emotion socialization, and then to look at group differences in adolescent brain function. So with that, it brings us to the Tuning into Teens brain study. And in this study, we try to answer two research questions. So the first, we wanted to look at whether there is a causal relationship. So does parent emotion socialization lead to changes in adolescent neural function during emotion regulation? And for the second question, we wanted to look at brain behavior relationship. So whether changes in the brain function were associated with reductions in adolescent internalizing symptoms such as depression and anxiety. For the experimental manipulation, we use the Tuning into Teens program. Many PAFRA members may be very familiar with this program, and it's developed by Sophie, who is probably in the audience as well. So Tuning into Teens is an evidence-based parenting program that has shown to improve parent emotion socialization and reduce adolescent internalizing symptoms. It teaches parents a supportive way of responding to adolescents' emotions called emotion coaching. To illustrate these 5 steps, I will invite you to imagine a young adolescent called Isabel. She auditioned for a role in her school play, but unfortunately she didn't get it. So for the first step, Isabel's parents may notice that she is a little bit quieter than usual. They take a moment to recognize something may have happened, even though Isabel may not have said anything yet. And for the second step, view emotions as opportunities to connect. Here, instead of dismissing her feelings, they see it as a chance to have a discussion about emotion with Isabel. So they can ask something like, I notice you're a little quiet today. Is everything okay? Isabel may respond with something like, I didn't get the role. I'm so sad. And rather than trying to jump in and fix the problem or tell Isabel that, don't worry, darling, you'll have other opportunities in the future, or you have tried your best, and we don't always get what we want in life. They could simply just listen and empathize with Isabel and say, that must be really disappointing. It means a lot to you. The next step, help adolescents name emotions. So this could be something like asking Isabel whether she feels frustrated at herself, or maybe she's feeling jealous of Emma, who seems always gets what she wants. So just help her to have more emotional clarity. And the last step is to go through problem solving, and we always encourage parents to involve young person in this step. Okay, so looking at the study design, so we have participants at the baseline where adolescents completed a brain scan, and both adolescents and their mothers completed some questionnaires. And then through the randomization, some participants will receive the Tuning into Teens intervention. And just to note that we use the one-on-one version of the Tuning into Teens, which is quite innovative as well. And then at the 6-month follow-up, we invite them back for the same brain scan and the same set of questionnaires. And this is the demographic of the study sample. As I mentioned briefly, we only have mothers and daughters in this study because it's a relatively kind of small study. So we wanted to keep the sample as homogeneous as possible. And we see that internalizing problems are higher among female adolescents than male adolescents. And the mean age of adolescents at baseline is 11.45, so within the early adolescent age bracket. And all the adolescents experience elevated internalizing symptoms at baseline. And here we can see that our sample is made up of participants who are from relatively high socioeconomic backgrounds, and we wanted to acknowledge that as well. Okay. Moving on to this page, it may look very crazy, but I do want to walk you through it, and I promise it's not as bad as it looks. So at the top, we have the 2 emotion regulation tasks adolescents did while they are in the MRI scanner. So the purpose of having these 2 tasks is to tap into 2 forms of emotion regulation. So one is more implicit, automatic form of emotion regulation, and one is more explicit form of emotion regulation. So for the affect labeling task, adolescents were just looking at the emotional phase versus they were asked to put in a label to the emotional phase they are seeing. So whether it's the angry phase or a sad phase, so they will use the bottom box to make a choice. So even though they are not kind of consciously aware of that they are regulating emotions, the act of putting feelings into words has shown to reduce negative affect. And then for the cognitive reappraisal task, they were shown a bunch of negative pictures. And for some of them, they were just asked to look at the pictures and let themselves feel whatever way they might be feeling. Versus for another subset of the photos, they were asked to come up with something to make them feel better. So looking at this picture, they can say it's from a movie, it's fake. Or they can say, oh, the girl looks really sad, but maybe her family, they are coming to help. So kind of try to come up with something to make them feel better about what's happening in the picture. And for the statistical analysis, I tried really hard over the weekend to come up with an example, and I hope this one works. So imagine you are hosting a dinner party. So the pre-processing step is when you are doing all the washing and chopping, getting the ingredients ready. So just as you can't cook the messy and unprepared ingredient, we can't analyze the raw brain data directly. So we need to remove the noise that might come from the slight head movements when adolescents are in the scanner, and standardize each person's brain image to the template so the brain regions align with each other. So for the first step, it's like when you start cooking. So you may saute the vegetable, grill the meat, boil pasta. So you focus on each part of the meal before putting them together. And in fMRI, what we are doing here is to look at how each participant's brain responds to a certain task. So it will be looking at the first participant and how their brain reacts when they try to label emotion versus when they just look at an emotional phase. So by doing this step, we get kind of a brain activity map for each participant. And the second level analysis is when we put all components together and serve the meal. So here we combined each participant's brain activity map and run statistical analysis using kind of regression, ANOVA, more traditional statistical analysis methods. And in our study, we use the flexible factorial design, which is basically a repeated measure ANOVA, where we look at which cluster of the brain is significant in this group by time interaction. And because the brain is made of hundreds and thousands of voxels, we need to apply correction for multiple comparisons. And here, the region of interest analysis is when we apply the predefined mask and we apply corrections to these regions that we that was implicated in emotion regulation. And we also do the whole brain analysis to see whether there are other significant regions. Okay, having that out of our way, looking at the results now. So for the results, we did find tint intervention leads to changes in adolescent brain function. So for the affect labeling task, we found a significant group by time interaction in the superior frontal gyrus. We then extract and plot each participant's brain activity in this cluster. And we can see in the plot here, the green line indicates the intervention group. So after the intervention, adolescents showed increased activity in the superior frontal region. And this may mean that after their mothers participated in tint and practiced emotion coaching, adolescents were able to engage prefrontal control regions more when they regulate emotions in a more unconscious and automatic way. And in contrast, when looking at results from the cognitive reappraisal task, so remember this is a more explicit form of emotion regulation, we see that adolescents in the intervention group actually showed reduced activation after the intervention. This may mean that maybe when they are instructed to reinterpret emotional stimuli and regulate their emotions, less cognitive effort is required when they are trying to regulate emotions in a more explicit way. So next, we wanted to look at the brain behavior associations. So I just wanted to go through this graph quickly. So the x-axis is the change in brain function. So this is when we subtract baseline from the follow-up scores. So when you see any values that's below 0, it means a reduction in brain activity at follow-up. And y-axis is adolescent reported internalizing symptoms. And again, a negative value indicate a reduction in internalizing symptoms. And of the two regions, we found significant group by time interactions. We showed that reductions in inferior frontal gyrus was associated with reductions in adolescent reported internalizing symptoms following the intervention. However, we didn't find significant associations for the superior frontal gyrus cluster, and we didn't find any brain symptom correlations for mother-reported adolescent internalizing symptoms. Now, bring it all together, just a bit of reflection before wrapping it up. As mentioned before, the sample is from high socioeconomic background, and we only included mother-daughter diets, which may limit the generalizability of our study findings. And it's also a question, it's kind of still a question in neuroscience as to how well and whether regulating emotions in an MRI scanner represent adolescents' day-to-day emotion regulation experiences. And kind of just reflect on the brain behavior relationship. We did find a association with the inferior frontal gyrus cluster, but not with the superior frontal gyrus cluster. And that prompts us to wonder that maybe this relationship will emerge with a longer follow-up, and these are things to consider. Lastly, we provided further evidence for the tuning into teens intervention, in that it not only has influence on the behavioral self-report outcomes, but it also impacts adolescents at a neural level. But however, I do know there are a lot of clinicians and researchers in the room, and I wanted to hear your thoughts about how we can best use neuroscience to inform interventions and how to best translate neuroscience findings into clinical practices in the future. Okay, and lastly, I just wanted to say thank you to everyone who are involved in the Teen2Be study, especially all the families, all the amazing kids, all the amazing adolescents participated in the study as well. Thank you.
Thank you, Sylvia. I really like your cooking example to explain fMRI. I might have to use that one day. All right, thank you, Sylvia. Again, we'll move on to Sarah's presentation. If you have any questions, note them down and we can come back to answer them after Sarah's talk. Thanks, Sarah.
Right. Oh, sorry, just give me a moment while this loads. Oh, sorry, everyone, having a bit of a technological issue. I'll be okay. There we go. All right.
Looks good.
Yeah, yeah, sorry, everyone. It's done it again. All right.
That was good.
Perfect. Well, thank you, Sarah, for the introduction, and thank you, Sylvia, for touching on a lot of the topics that I will also be touching on. So as Sarah Whittle mentioned, my name is Sarah. I was one of the PhD students at University of Melbourne that worked with Professor Whittle and also Professor Marie Yap. And today I'm going to be discussing some findings relating to adolescent brain structure and whether it mediates the association between maternal and paternal parenting behaviours and adolescent internalising problems. So as Sylvia has mentioned, and as most of you probably know, internalising problems are extremely prevalent in mental health issues for children and adolescents globally. And internalising problems typically capture depressive problems and anxiety problems. But literature has identified that genetics contribute approximately 30 to 60% of the risk of development of these mental health problems. And that suggests that the remaining gap may lay within environment. Now, the environment is extremely broad, but they are particularly important to consider because unlike genetics, which can be quite difficult to change, the environment typically, the early environment can be quite modifiable, which means it might be receptive to prevention and intervention. So within that environment, parents play a really instrumental role in child and adolescent development. And as Sylvia mentioned, they are the primary source of socialization and rearing, and they often lay the foundation for the parent-child relationship. They determine the child's security with their caregivers, and they often model behaviors and emotional responses in particular contexts. So a child will often learn normal responses and reactions to certain situations and also shape their schemas and beliefs about the world around these early experiences and modeling behaviors. And these beliefs and processes may have links to their later mental health problems, particularly if parents are modeling fear, negative affects, or maladaptive parenting behaviors. It's also very likely, however, that parenting influences a child or adolescent's mental health as the result of the influence on their brain development. So as brain development is undergoing a lot of change and reorganization during adolescence, particularly being extremely plastic at this period, as Sarah mentioned, it has been suggested that adaptive or positive parenting behaviors may support normative brain development, while maladaptive or negative behaviors may alter or disrupt brain maturation. So this might be particularly relevant to areas which underpin emotion regulation, fear, and decision-making, again, as Sylvia previously described. And this may cause difficulties with processing stress, emotions, and other challenging events, with literature often implicating the amygdala, the hippocampus, and prefrontal cortical regions such as the anterior cingulate and orbitofrontal cortex. So non-normative changes within these regions have been suggested to play a role in the onset of internalizing problems such as anxiety and depressive symptoms, as they often hinder the development of emotion regulation skills. And while exposure to these environmental and more specifically parenting factors are likely to impact development from infancy all the way to young adulthood, adolescence is a period which sees remarkable change to brain structure and is often a period where we see the first emergence of mental health symptoms or disorders. Now, there are numerous studies that have already touched on the association between parenting behavior or even parental mental health problems and adolescent brain development or internalizing problems. But as you can see, fathers are highly underrepresented. So there are still very strong, resounding gender stereotypes and social perceptions of gender roles, which see mothers often at the focal point of this literature. And some have gone so far as to suggest that paternal caregiving is more congruent with friendship to children, with others suggesting that it's so similar to mothers, it sort of justifies their absence in the literature. Now, obviously, we know that emerging literature is suggesting that paternal parenting behaviours may actually hold differential effects than that of mothers. And it's really important for further investigation to continue. However, with that being said, I of course want to acknowledge the fact that the mother-father dynamic is not the only family dynamic that requires investigation. And of course, there's a plethora of different family structures and environments that are equally as important. But as paternal roles and paternal caregiving has been grossly overlooked in the existing research, we wanted to investigate paternal and maternal interactions and their interplay therein as a first step to broadening this area of research. So the general aims of this current study were to investigate whether independent and interactive roles of maternal and paternal parenting behaviours, specifically acceptance, and its relation to adolescent structural brain development and mental health outcomes. So we investigated the role of brain structure as a potential mediator to determine whether the changes in the brain might play a role in the effects of maternal, paternal, or the interaction of maternal and paternal acceptance in adolescents' mental health symptoms. Now, to do this, we used the data that exists from the Adolescent Brain and Cognitive Development Study. So for those who are not familiar with the ABCD study, this is a study that is currently ongoing in the United States of America across 21 research sites. It has recruited approximately 11,800 youth, and it intends on following them across 10 years. So from around age 9 to 10 to around age 19 to 20. Now, because our sample specifically was looking at individuals with a maternal and a paternal caregiver, whether that was a step, adoptive, or biological maternal or paternal caregiver, our sample resulted in 2,908 adolescents to investigate. The benefit of this sample is also that it is intentionally diverse in terms of representing race, and it represented them as currently in the American community. Now, the factors that we investigated in this study were parental acceptance, which was rated by adolescents at around 10 years old. And it is important for me to note that acceptance is the higher end of this scale, but low acceptance can be seen as the equivalent of rejection. We also investigated the amygdala, hippocampus, the caudal and rostral anterior cingulate, and the lateral and medial orbitofrontal cortices at year 2, which when children were or adolescents were around 12 years old, again due to the prevalence in their role in internalising and their role in the literature and how they've been investigated in emotion regulation. And we investigated adolescent internalising problems in the preliminary analyses when adolescents were at year 3, so around 13 years old. But we did also conduct supplementary analyses on specific depressive symptoms and anxiety symptoms as well to determine whether there were any differences there. And of course, I will note that in adolescent symptoms were reported by the parent, not the adolescent themselves. So to give an overview of the model that we are using to look at the relationships, it all looks a little bit complicated, but I will break it down. But we use structural equation modeling, and this allows us to sort of lightly infer some causality between the variables. And at first, we wanted to determine whether the findings were different or similar for adolescent biological sex. So there is evidence that males and females at that period of development have differences in brain development and also symptoms. As we know, females are often higher on symptoms at an earlier age. And the results did determine that males and females were quite different. So all analyses were conducted separately. We also controlled for the symptoms at baseline as well as brain structure at baseline for the regions I mentioned. And that was to help us determine change across time that could be the result of the parental factors. So our preliminary findings indicated that paternal acceptance was directly associated with reductions to depressive symptoms and internalizing symptoms for male adolescents, with no findings there for maternal acceptance for young males. We also found that for mothers' acceptance, there was a direct reductive effect on female adolescent depressive symptoms. So we're seeing a little bit of a trend there with maternal acceptance associated with daughters' symptoms and paternal acceptance associated with their sons' symptoms. However, we did also find that there was an interaction between maternal and paternal acceptance, which also reduced female adolescent depressive symptoms. Now, it is important to note that we did not find any relationship between maternal or paternal acceptance or the interaction therein and adolescent anxiety for males or females. But returning to that interaction, we see that on this graph here, so it is a little bit difficult to interpret, so I'll try and ensure that everyone kind of grasps the concept. But more plainly, we see that there is a slope where we see the trajectory of depressive symptoms. Now, what this graph is telling us is that when maternal acceptance is high, depressive symptoms are already low for young females. However, when paternal acceptance is also high, there's a compounded effect whereby female depressive problems reduce at a more rapid rate. So there's a strong relationship with having two parents exhibiting that acceptance behavior. Now, in regard to the brain areas that I mentioned, we see that maternal acceptance has a reductive effect on grey matter, so one facet of brain structure. So we see maternal acceptance up the top there is associated with a reduction to female caudal anterior cingulate volume. And we also see maternal acceptance related to male rostral anterior cingulate, lateral orbitofrontal cortex, and medial orbitofrontal cortex volumes. So reductive effect, but all coming from maternal acceptance. Paternal acceptance was surprisingly not related to brain volume for grey matter for either sex, males or females. So all of these associations were directly coming from maternal associations. We also see for subsequent analyses, so for cortical thickness, again, we're seeing maternal acceptance at baseline or year 0, having a reductive effect on male rostral anterior cingulate and medial orbitofrontal cortex cortical thickness. Again, no findings were determined between paternal acceptance and cortical thickness for either gender. And then we see the cortical thickness associations with mental health, whereby caudal anterior cingulate for females was associated with reductions to internalizing symptoms at year 3, while the lateral orbitofrontal cortex cortical thickness was associated with an increase in depressive symptoms in males at year 3. So the thickness of those regions had a direct relationship with the symptoms of male and female adolescents for those regions. In regard to surface area, so surface area of the caudal anterior cingulate and rostral anterior cingulate for females was directly related to anxiety symptoms. So I'll get you to reflect on the symptoms that I mentioned previously, whereby male and female adolescents neither had associations with paternal or maternal acceptance for their anxiety symptoms. But we see here that there are brain regions that are having effects on the symptomatology relating to anxiety symptoms. And the same can be said for female adolescent depressive symptoms, where we're seeing all of those prefrontal cortical regions having a negative relationship with depressive symptoms, suggesting that the surface area of these brain regions has a reductive effect on depressive symptoms. And finally, for internalizing symptoms for males and females, again, for females, we see all of those prefrontal cortical areas reducing the intensity of internalizing symptoms for female adolescents, and as well for the anterior cingulate for our male adolescents. So we can see the surface area of our caudal and rostral anterior cingulate has a negative association with internalizing symptoms. So to summarize these findings, what do they actually mean? Now, our findings did not identify anything between parental acceptance and the amygdala or hippocampal volume, surface area, or cortical thickness. So we didn't find any significant associations with those regions, neither for the relationship between the amygdala and hippocampus and anxiety, depressive, or internalizing symptoms for either sex. Now, given that grey matter volume is defined by a calculation of surface area and cortical thickness, it is possible that a lot of these relationships that we saw from brain region to adolescent symptoms are driven by surface area at this age, but it might have a more profound impact on development of internalizing symptoms at this age, so from the age 9 to approximately 13, although this is speculative. Now, there was also no evidence to suggest that grey matter volume, cortical thickness, or surface area, so generally brain structure at these ages, could affect or could mediate the effect of maternal or paternal acceptance on any of these outcomes. So essentially what that means is brain matter volume or brain brain structure at this age did not change the effect of maternal or paternal acceptance on the outcomes that we saw. It is likely, however, similar to what Sylvia mentioned in her study, that the time span between year 0 and year 3 was too short to see a meaningful amount of change in brain development to determine any associations, because obviously this is the very beginning of adolescence. So some research has suggested that the volume and cortical thinning of the anterior cingulate cortex and orbitofrontal cortex are associated with heightened depressive problems for adolescents and adults, which we did see emerge in our study. Although findings for surface area in this study were somewhat novel and require a bit more further investigation. Now, again, we did see some findings for same-sex parent-adolescent dyads. So quite interesting to see how paternal caregiving, or in this instance, acceptance, was directly associated with mental health outcomes for their sons, while mothers' acceptance was directly associated with mental health outcomes for their daughters. So this might suggest that there are specific relationships between mothers and daughters and fathers and sons, whereby the parenting behavior exhibited is directly influential to the same gendered child. Or it might suggest that acceptance specifically is a behavior that is directly associated with the gender shared between parent and adolescent. Now, we did also note that fathers' parenting differed to mothers in prediction of brain development. So it's quite interesting to note that finding. Now, it might suggest that due to maternal early life rearing or time spent between mother and child, that there is a more profound effect of maternal parenting on the development of the brain. However, it might also suggest that this is a specific finding for parental acceptance, and that maternal acceptance is directly associated with changes to the brain structure, but then alternate parenting behaviours might show the same results for paternal caregiving. And lastly, we did see some similarities and some differences, but we also noted that parental acceptance does interact and augment the trajectory of depressive symptoms for young females. Now, of course, there were some limitations to this study. We only included acceptance or the lower end of acceptance, which is the equivalent to rejection, and we did not investigate parental mental health in this study. We were also very, very specific with the regions that we investigated, being such a novel study, and there's definitely likely other regions that would impact these or be impacted by parenting behavior and likely impact the trajectory of mental health. But more importantly, there are other family factors that are very important to consider, such as interparental conflict, parent-child attachment, culture, race and ethnicity, custody, divorce, and parent time allocation, and also bidirectional associations. So to just sort of pinpoint 2 out of those 5, culture, race, and ethnicity shows us that parenting behaviors can actually have different associations with their adolescents because the perceptions can be different based on culture or race. So some parenting behaviors can be seen as adaptive in some cultures and maladaptive in others, and it's really important to take these cultural differences into consideration before making generalizations. And lastly, with the bidirectional associations, some literature has found that when children or adolescents are experiencing mental health problems, the parenting behaviors and that are used by parents do often change. They can become slightly more protective, use higher monitoring, and it can also affect parental mental health. So it's important to acknowledge that this is not about identifying blame or, suggesting that parents have a unique and the sole role in the brain development of their children or mental health, as Sarah mentioned at the beginning. But it is important to understand that the family unit, there are a lot of associations therein, and there's a bit of a spillover effect where the mental health or behaviour of one can affect others, and that works both ways for parents and children. But lastly, the implications. Paternal caregiving is highly influential to male adolescents, but maternal caregiving in this instance was found to be highly implicated in brain development. Parenting behaviours were found to interact, so it's important to consider that if a child has two parents or two caregivers, the parenting behaviours do not occur in a vacuum. The child is not likely to be experiencing one parent's behaviour and the other parent's behaviour and not process them simultaneously. So it is important to consider the larger picture and the more holistic family environment. Parent and family interventions should engage both or all caregivers, whether that be two parents or four parents if there's step parents involved, as we can see that the interactions and the differences can impact the adolescent. And there are differences for internalising depressive and anxiety problems, so we should also be wary of those differences. But most importantly, I think this is a first step to acknowledging that fathers are just as important as mothers in their child and adolescent development. And more research certainly needs to be done with both caregivers, but they also need to be represented in interventions, and this should have consequences or considerations at least for policies such as paternity leave and custody arrangements, as we see maternal caregiving might have more implications for brain development. Again, we need to do further research on this and determine whether it is due to early lifetime or bonding, but it certainly suggests that fathers should and deserve to have a similar opportunity to do so. And hopefully, if you do have any further questions or you would like to read up on a meta-analysis that we conducted that sort of breaks down multiple parenting behaviours for fathers, mothers, and adolescents and children, please scan that QR code there. But thank you all so much for your time. Of course, I'd like to thank Sarah and Marie and the lab that I worked with previously. And just a reminder that ICEP's deadline for abstracts for next year is coming up on November 15th. But thank you all for your time.
Thank you, Sarah. Thank you both. Thank you everyone for sticking with us. If anyone has any questions, either specifically for Sylvia or Sarah or more general questions, please feel free to either kind of put your hand up or just unmute and ask, or feel free to pop it in the chat and I can read it out. Anyone have any questions? No questions? All right. Sarah, could you pop up the QR? Oh, okay. Sorry. Can we put the QR code up again? And there's also a question for Sarah.
Yes.
Did you look at whether the parents were working full or part-time and their availability for their children?
Yes. So unfortunately, this wasn't something that we specifically investigated, but we absolutely acknowledge that that is is super important. Having looked at this literature, I suppose some people investigate parent and time allocation in custody arrangements, but it's also important to determine quality time. A child might have shared custody, but that parent might be working two jobs. So it's certainly important to understand not only the custody arrangement, but also the actual time that the parent and child can spend together when they they are together. So no, unfortunately, we didn't look at it in this study, but it's definitely something I am considering for future research.
Thanks, Sarah. Any other questions or comments? Would anyone like to address Sylvia's question, especially, I guess, clinicians in the room potentially, or clinical researchers? Any thoughts on how this kind of neuroscience research could be helpful for, or kind of translated into the clinic?
Yeah, I would appreciate if there's any comments or suggestions on that.
Okay. Thank you everyone again for joining. Thank you, Sylvia and Sarah. Now, a recording of this seminar will be put up on the PAFRA website in the coming days, pafra.org/seminars. Yeah, with that, thank you again everyone for joining, and hope to see you at the PAFRA, or the next PAFRA seminar. Okay, thank you.
Thank you.